Evolutionists who believe in the accidental emergence of life on early Earth have put forward many hypotheses to explain the origin of life. According to them, all the species we see today evolved from one single component of a living cell that arose by chance from non-living matter. They focus primarily on three major components in a living cell: DNA, RNA, and proteins. DNA is an extremely complex nucleic acid that contains all the information for building and maintaining living beings throughout their lifespan, especially the information about manufacturing proteins. RNA is another extremely complex nucleic acid that carries instructions from DNA to the ribosome for making proteins, and proteins carry out different tasks and catalyze all biochemical reactions essential for survival. The evolutionary researchers of the origin of life initially claimed that proteins were the first macromolecules to appear on early Earth. However, after failing to obtain proteins even under highly controlled lab conditions and realizing that proteins cannot be built without pre-existing proteins and DNA to guide them, their attention shifted to RNA as an alternative, which later came to be known as RNA World Hypothesis. It was first suggested in early 1960s and was advanced by Walter Gilbert in 1986. According to this hypothesis, RNA was the first to emerge in the primordial Earth. It then somehow replicated itself, began to synthesize proteins, later felt the need to store information more stably and thus evolved into DNA, and then this way eventually formed a whole cell. However, no research has ever proven how such a molecule with an extremely well-designed structure could have come into being in the early Earth's random environment, and how it would have survived and replicated under harsh and unprotected conditions of the early Earth, especially considering that RNA is prone to heat, unstable, and degrades quickly. Even the second law of thermodynamics states that under random natural conditions, instead of forming spontaneously, complex organic molecules are way more likely to fall apart. So there is no way these molecules could have emerged by chance, but even if we suppose that first RNA appeared randomly, it alone would have been of no use. One of the founders of the RNA World Hypothesis, Leslie Orgel, also acknowledged this fact that for the prebiotic RNA to work, it would require two properties absent in modern RNA: the ability to replicate without the assistance of proteins and the capacity to catalyze each step of protein synthesis. So far, all the experimental attempts to validate these requirements even in the lab turned out to be problematic in one way or another. One of the experiments conducted on RNA for self-replication without the help of enzymes in 2016 by Jack Szostak, a Nobel Prize winner, was later found to have misinterpreted results, for which reason they had to retract their research. The researchers admitted that their strong belief in the hypothesis had blinded them to the errors and that they should have been as careful as the person who repeated the experiment and yielded mismatching results. The fact that no mechanism for RNA self-replication without the need for enzymes could ever be created or found is a critical point that undermines the RNA World Hypothesis. Yet the belief is still strongly advocated and even published in textbooks as if it's a proven scientific fact. When RNA cannot self-replicate, it invalidates the whole argument for evolution. It is also noteworthy that even for the formation of RNA in biological systems, pre-existing RNA is required. RNA polymerase is a protein-based enzyme that reads the DNA code and synthesizes mRNA. In other words, RNA cannot form without an RNA and proteins already present, posing a great difficulty for evolutionists which they have never been able to explain in terms of evolutionary developments. While RNA does carry genetic information from DNA, it cannot synthesize proteins without the help of other proteins. And to form proteins, the presence of other proteins, DNA, RNA, ATP, a controlled protective system, and a complete cell must all exist simultaneously. The entire system is so interdependent that one cannot form or work without the other, and it only makes sense if all of these components were created at once. In fact, Leslie Orgel himself pointed out in another paper that this interdependent system might even mean chemical evolution just never happened. This scientific reality aligns perfectly with the creationist view that God the Almighty created a complete cellular system that requires all its organelles to be present and functioning simultaneously. That's why we see only perfect beings around us, not any transitional form. The failure to demonstrate the accidental emergence and replication of RNA even after 60 years of research and experiments has only highlighted the impossibility of such complex biomolecules arising by chance on early Earth. Yet evolutionists without any scientific basis remain profoundly attached to this idea, simply because they seek a different explanation for the existence of life even when it goes against the true scientific evidence.